Hello. Okay, now we have stopped somewhere here in our previous class where we discussed about the endopeptidases and the exopeptidases. So today let us continue with that, and here we are going to see how the digestion of the protein takes place in the intestine. Recall, we have said that there are different enzymes that are secreted by both pancreas and the intestinal glands. Okay. Now trypsin, chymotrypsin, carboxypeptidases, dipeptidases, tripeptidases and amino peptidases are different enzymes that are involved in digestion of protein in intestine. Okay. Now here we have on this slide names of different enzymes and activation. In my previous class I said that most of these digestive enzymes, particularly protein digesting enzymes, are present in their inactive forms to prevent their adverse effect on the tissue. Okay. Similarly, trypsin, chymotrypsin are also present in their inactive form. The inactive form of trypsin is called as trypsinogen, of chymotrypsin is called as chymotrypsinogen, and carboxypeptidases is called as procarboxypeptidase. Similarly, there is another enzyme called as elastase that will be present in the form of proelastase. Now, when the food is there in the intestine for its digestion, what happens? This trypsinogen must be activated to trypsin, and there comes to picture one of the major enzymes, okay, that is called as enterokinase enzyme. Okay, enterokinase enzyme, and remember it is secreted by small intestine. Okay, this enzyme is secreted by small intestine, and this particular enzyme, it does not have any digestive enzyme property. Okay. Instead, its work is to activate other enzyme, particularly it activates trypsinogen. This enterokinase enzyme activates trypsinogen to trypsin, okay, and this happens in alkaline pH. The pH of intestine is alkaline, pH of stomach is acidic. That is why food, when it comes to the intestine, will be carrying pepsin with it, but it gets deactivated because of the alkaline pH. Recall, we said that any pH above 5, pepsin gets inactivated. Okay, but trypsin will be active in its alkaline in the alkaline pH. So, enterokinase will activate trypsinogen to trypsin. Now, again trypsin has a autocatalytic property, and it can itself digest the remaining trypsinogen to trypsin. Not only that, the same trypsin can also activate chymotrypsinogen to chymotrypsin and can also activate procarboxypeptidases to carboxypeptidases. So, here we are saying the activation of 3 different enzymes by one single enzyme, that is trypsin. So, in the beginning trypsinogen gets activated by enterokinase enzyme. These 3 enzymes what we are discussing are the secretions of pancreas, whereas enterokinase is the enzyme of small intestine. Anyway, by the end of the activation, the system will have trypsin, chymotrypsin and carboxypeptidase. So, here we have another picture which can help you to understand how the activation takes place. So, enterokinase will act on trypsinogen, converts it into trypsin, and this trypsin again activates trypsin, and activated trypsin will activate chymotrypsinogen to trypsin. Okay. So, then proelastase to elastase, procarboxypeptidases to carboxypeptidase A, and again B to carboxypeptidase B. This is how the activation of different enzymes takes place in the lumen of the small intestine. Now, once trypsin is ready, okay, after the activation, it will act on the proteoses and the peptones. Recall, in stomach, protein digestion has resulted in the formation of proteoses and peptones, and trypsin will act on those proteoses and peptones to generate dipeptides, tripeptides and polypeptides. Okay. Similarly, these chymotrypsin are also, they are involved in digestion of proteoses and peptones, and they generate di, tri and polypeptides. Dipeptide means when there are two amino acids joined together by a peptide bond. Tri, three amino acids are joined. Imagine a small chain with the two beads, a chain with the three beads like this. Polypeptide means a whole chain of beads. Okay. Chymotrypsin, okay, and these two are specific because this trypsin it cleaves in a place wherever arginine and lysine are present, whereas chymotrypsin will break wherever phenylalanine, tyrosine, tryptophan or leucine is present. These are all the names of amino acids. So, wherever these amino acids are present, specifically they will cut the particular polypeptide chain. And recall, these enzymes are endopeptidases. Okay, these are the endopeptidases. Trypsin, chymotrypsin are endopeptidases. Okay, whereas carboxypeptidase and the rest of them, dipeptidase, tripeptidase are all exopeptidases. Okay, yes. Now, so by the end of the activity of trypsin and chymotrypsin, you see di, tri and polypeptide formation. I am repasting the diagram here so that you can have a very clear idea. Endopeptidases and the exopeptidases. So, here you see this is where actually trypsin and chymotrypsin are, and this is where carboxypeptidases and rest of the peptidases act. Okay. Yes. Next, moving on with the digestion process. So, here now what is left in the intestine? All the protein was first digested to to proteoses and peptones. They were digested by trypsin and chymotrypsin to di, tri, polypeptidases, peptides. So, now what happened? These two should be further digested into smaller units, amino acids, okay, or di or tripeptides, so that it can be easily absorbed. And that work will be done by the carboxypeptidases. Okay, A and B will help that. Okay, carboxypeptidase will act on these di-tri-polypeptides and convert them into amino acid, the simple forms of proteins or simple monomeric units of proteins. Okay. Next one, then there are also dipeptides. They will be acted upon by these dipeptidases. Dipeptides will be acted upon by the tripeptidases. Okay, and there are also amino peptides. Those contain amino groups. They will be the region that contains amino group. So here you can see. So amino peptidases, they cleave this particular bond. Wherever there is a amino group, that will be broken by amino peptidase. Okay, and if it is a terminal one, carboxypeptidases can cleave them, so they can separate them. So these will be separated as amino acid, individual amino acids. Okay, so that means at the end of this digestion, we'll have what we'll see are some free amino acids and some dipeptides and a few tripeptides will be present in the digested food. Okay. So, this is how the digestion of food takes place in the intestine. Now, we have completed the digestion of protein. So, to recall, digestion of protein takes place in the stomach and in intestine. Stomach by the help of pepsin and intestine by the help of trypsin, chymotrypsin, carboxypeptidases, amino peptidases, dipeptidases and tripeptidases. Okay. So, by the end of protein digestion, we have amino acids, few dipeptides and few tripeptides. Let us see how the absorption takes place. Absorption takes place in the small intestine itself because there are microvilli, finger-like projections that are present on the epithelial cells. Okay. Now, let us see how the absorption takes place. You are familiar about absorption. Absorption is the process of transfer of digested food material through the intestinal wall into the circulatory system so that it can be used up by different cells. Now, first amino acid absorption. This amino acid absorption, it is a sodium dependent co-transport absorption. Recall glucose absorption in carbohydrate digestion. There also it is a sodium dependent glucose transfer process or co-transfer process. Similar process occurs here too. So, it is an analogy of glucose co-transporter, and here there are actually 7 different carrier proteins to different groups of amino acids. There are different classes of amino acids like basic, acidic, neutral amino acids, etc., amino acids and all. So, based on the class of amino acid, there are 7 different types of co-transporters. They help in transport of amino acids from the intestinal lumen into the cell of small intestine. From here it will go to the blood. Okay. So, now let us see what happens here. Again, like in case of carbohydrate digestion, sodium that is present in the lumen will be first released out to the outside, not in the lumen, sorry, in the cell. It will be taken out by sodium potassium ATPase pump. So, it is an active process. Now, next what happens, sodium binds here to this protein co-transporter system, and then the amino acid, specific amino acid also binds, and by this binding then both of these will be then taken into the cell. So, the now amino acid is present, sodium is also present. This amino acid will be then transported by facilitated transporter system. There are 4 different such amino acid transporters, okay, present in the basal membrane of the epithelium, which will transfer the amino acid to the portal vein. Okay, and this sodium that has come will be pumped out, so that some concentration of sodium is always maintained, okay, within the cell, so that transport is easier. So, this is how the amino acid is absorbed, okay, into the cell. Now, see, this is what I have already said. There are 4 different types of carriers are present. They are going to transfer to the next part of the from the epithelial cells of the intestine to the circulatory system, they are passed. Next one, we have dipeptides and tripeptides. So, it is not necessary that all the peptides should be broken down into amino acid. Even di and tripeptides can also be absorbed by the intestinal cells. What happens here? Let us see. So, these dipeptides and tripeptides are absorbed by a special type of co-transporters called as proton dependent co-transporters. See, amino acids are transported through sodium dependent, here they are transported through proton dependent co-transporter systems, and there is a special name for it. It is called peptide transporter 1. So, here you can see peptide transporter 1. And in this process what happens, I think you can just concentrate on this. In the beginning, sodium will be it will be taken into the cell, and proton from the cell is taken out to increase its concentration here in the lumen. Okay. Then what happens, once the concentration has increased here, next the dipeptides and the tripeptides will be passed in through this peptide transporter 1 together with the proton. So, that is why it is called as a co-transporter system. Okay. It is a proton dependent co-transporter system. So, proton also comes in, and the dipeptides as well as the tripeptides will also come into the cell. Now, here you see proton concentration will actually increase here, proton, because of the entry, as well as di and tripeptides. These di and tripeptides will be further broken down into amino acid inside the cell itself by the help of enzymes, peptidases that are present in the cell. Now, there is also one more thing. We saw that the protons have entered in by this process. These protons will be thrown out by this particular transporter. It is an antiport system. Antiport is system where the ions are moving in the opposite direction. So, sodium is taken in and potassium is pumped out. a proton is pumped out here. Okay, we'll just stop the story somewhere here itself. Let's continue with our amino acids. So now see, dipeptides and tripeptides are converted into amino acids, and these amino acids are then sent to the circulatory system through these transporters that are present in the basal layer of the cell, okay, epithelial cell. Okay. Now we are left with something else. This sodium ion is present inside. This will be thrown out of the cell, okay, so that continuous inflow can take place by this ATPase that is present in the basolateral membrane. I am sure it is confusing, so let me repeat what we have left in the previous one. So, free amino acids will be taken into the cell by sodium dependent co-transporter, where sodium and amino acids are pumped together into the cell, and then that amino acid is sent to the blood. This process of taking in the nutrient into the cell of the intestine and from there to the circulatory system is called as absorption, isn't it? Okay, so here sodium and amino acid come together, then amino acid is taken into the blood, sodium is pumped out, okay, so that it is now available for further circulation, and this is done by the utilization of energy. So it is an active transporter system. Whereas when you come to the absorption of dipeptides and tripeptides, it is slightly different, because here proton is involved and that transporter is called peptide transporter 1, okay, and here to maintain this proton, in the beginning proton will be pumped out of the cell to the lumen, okay, and this cannot be done easily. It should be done with the help of an antiporter, where for the number of protons pumped out, number of sodium are taken in. There will be excess of sodium here. To maintain the balance, this sodium is thrown out, potassium is taken in by the help of ATPase. And here this proton will be then helped, or this helps in the inward movement of dipeptides and tripeptides by the help of a co-transporter. When this dipeptide tripeptide comes into the cell, it is broken down, okay, it is broken down into, it is broken down into amino acids, and those amino acids are then taken into the cell, okay. So here we have one more picture. I think let me just zoom it and show you very clearly. So dipeptides and tripeptides are present here, okay, you can see there. So by the help of peptide transporter, it is taken in, okay. Cytosolic digestion takes place here. By the help of amino acid transporter, amino acids are taken into the blood, and this is the ATPase transporter through which the sodium is pumped out and potassium is pumped in, okay. So that's how the absorption of amino acids, dipeptides and tripeptides occur in the cell. And just a bit of additional information, the absorption is maximum in duodenum and jejunum. A very little amount of absorption takes place in the ileum, that is the last part of the intestine. So in this chapter we have seen proteins are formed of amino acids, enzymes that digest protein are called as proteolytic enzymes. Pepsin is the enzyme secreted in stomach, helps in protein digestion in stomach. Trypsin, chymotrypsin, carboxypeptidases, amino peptidases, di and tripeptidases are the enzymes of protein digestion in intestine, and absorption of amino acid takes place in the intestine, particularly in jejunum and duodenum area, and here it occurs by co-transport as well as by facilitated diffusion. And these are my references for today's class. Thank you.